For years, there's been interest in using off-the-shelf products, and iPSC-derived products, are attractive for one simple reason that iPSC, these induced pluripotent stem cell-derived either CAR T cells or CAR macrophages or CAR NK cells, you can produce a lot. You can create master banks of genomically defined iPSCs, and you can produce theoretically unlimited quantities of any cell product you want, and that's a huge advantage in terms of cost mitigation, accessibility, and the ability to genomically edit this with multiple edits, because you can make as many edits you want in the master cell line, and then in the iPSC derived stem cells, and then you can then produce the product of your choice with those gene edits. So not only does it allow the production of large quantities and potentially at a way lower cost, because you don't need to perform those gene edits every single time. You just need to produce in the master bank and then produce any cell you want. Not completely simple, because of the fact that you still need several weeks of manufacturing, and that does involve cost, but nonetheless it allows you really truly off the shelf production. And in this particular study, which was recently published in Lancet, it's an industry-sponsored trial by Fate Therapeutics, and they use CD19 CAR NK cell derived from iPSC, which also incorporated some other edits, including CD16, which is a very activating receptor in NK cells. So normally CD16, which binds to the Fc part of of IgG1 molecules, and is important for meeting ADCC, which is the antibody-dependent cell cytotoxicity, gets shared. Actually, my work back in my postdoc days, we found that CD16, every time it engages a monoclonal antibody coated tumor, it gets actually clipped. That's the downregulation mechanism. And following that work at University of Minnesota, they identified the exact sequence which is clipped by this enzyme called ADAM17, and they did a shared version, or the shared resistant version of CD16, and that's the version which was incorporated by Fate Therapeutics into their iPSC-derived NK cells, and that they made it enhance ADCC function. So this is the first in-human clinical trial of CAR, CD19 CAR with these edits, including membrane-bound IL-15 to enhance their anti-tumor function and survive in vivo survival, and had CD16. So really novel product. And it's a truly allogeneic product because it's off the shelf, and also it's not patient, it's not per patient, it's a great to use the master bank. And the what was exciting was the safety again. Despite the fact that these are allogenic products, none of those patients develop any graft versus host disease. And if you look at the response rates, they were not as good as use of autologous CAR T cells, but these are allogenic products, and use of allogenic products with all the challenges, I think the response rates were not bad. I think it can only get better, and that's the exciting part. This establishes the kind of baseline, if you will, efficacy signal of use of iPSC-derived CAR NK cells, and can only get better from there. And again, if you look at the in this trial, especially the patients with non-aggressive diffuse large B-cell lymphoma, like non-diffuse large B-cell lymphomas, like follicular and mantle, the survival and response rate were pretty good. So I think this trial tells us that it's feasible. The use of iPSC derived with multiple gene edits is feasible, it's safe, and has an efficacy signal which can only be improved from there, which is exciting.